Is this on? Yeah. Can people hear me? So I want to thank all of you for coming to Research Conference today. For those of you who I haven't met, I'm John Alexander. I'm a cardiologist here at Duke and at the DCRI. And Uptal has given me the privilege of introducing our speaker today. I have two stories to tell first. First, I was rounding in our cardiology intensive care unit last week, and we had a patient who came in with thyroid toxicosis. And she was pretty sick. She had, this speaks to all of the activities of thyroid hormone. So she was sick. She was hypotensive, dehydrated, tachycardic, had heart failure because her heart had been beating fast for a long time. She had some renal failure, mostly I think because she was dehydrated, and but she also had great lipids. Her lipid profile was excellent, and so what we're going to hear about today is some of the effects of thyroid hormone, I think, and how one can develop drugs that have some of the beneficial effects of thyroid hormone, but hopefully not all the effects of thyroid hormone, including those which are harmful. So I want to introduce Becky Taub. She's our speaker today. She's the founder and CEO of Madrigal Pharmaceuticals, and they have a thyroid receptor antagonist, a thyroid receptor beta antagonist. Agonist, sorry, thank you. We'll learn, well, I'm learning too, which has hopefully all the beneficial effects of thyroid hormone, but not the harmful ones. Becky lives in Philadelphia, and we actually met in probably 1992 when I was a medical student at Penn, and she was a genetics professor there. So she has been through a variety of pharmaceutical companies, both big and now as the founder and CEO of Madrigal, a small company that has a number of drugs in development in the cardiovascular and metabolic space. So she's going to tell us a little bit about this thyroid receptor agonist today and about Madrigal. So Becky.
Thanks very much. So it's really a pleasure to be here, and enjoyed my visit here. And I've been here before when Grendel brought us by here a few years ago when we were working on a different program for cardiovascular disease, and thought it was a really great opportunity to come by again and talk about the thyroid agonist, which we are calling 3196 for want of a better name so far. And Grendel, sort of made that happen, and then it turned out that this drug also has an amazing effect in the liver and a potential for treating non-alcoholic steatohepatitis. And I discovered an old friend, Anna Mae Deal, who was heading up this area, and Manal and Richard, who I've met with now, who are running a Nash clinical research network here. So it's like, it's been a great visit, and connecting the areas of cardiovascular and thyroid, endocrine, and hepatology as well. So I'll see if I can make some of those connections for you today, and I really invite all of your thoughts and questions and discussion as well. So what I'm focused on here is really how this is a great opportunity for what we call diabetic dyslipidemia, and potentially for NASH as well. And as you heard from John, Madrigal is a very small company. It's a virtual team. We have a few internal people, and then we work with a network of consultants and clinical research organizations. Our founder supporting operation is Bay City Capital, which is a venture firm in San Francisco. But as I said, we're located outside Philadelphia. The snapshot of this beta agonist is that we've completed phase 1 studies in humans with the compound, and demonstrated in a 2-week study in healthy volunteers up to 30% LDL cholesterol and 60% triglyceride lowering. with very excellent safety at all doses. We believe this is a best-in-class thyroid agonist with clear safety advantages over its previous analogs. And we're going to show probably more preclinical biology with this molecule than it's going to be a little bit complicated, but hopefully I can explain the preclinical biology and how it supports the idea that this compound might be efficacious in NASH. And so what we have here is a drug which has an opportunity in both dyslipidemia, particularly diabetic dyslipidemia, and liver disease. And we also then have preclinical programs in another metabolic target. And this shows our pipeline. I did want to highlight, I guess I'm supposed to use this pointer, that there is an orphan opportunity for the drug as well in a rare disease called resistance to thyroid hormone, in which there are mutations in the beta receptor or the very gene that this compound targets. So if we look at the cardiometabolic disease spectrum, of which you're all familiar, obesity, genetics, environment predisposes to a variety of different risk factors for cardiovascular disease and diabetes, and in this case, NASH and cirrhosis. And these risk factors include dyslipidemia, insulin resistance, and I would include in that non-alcoholic fatty liver disease, or NAFLD, which then can lead to the more severe cardiovascular and metabolic diseases. And this beta agonist has a potential to work at multiple points in this pathway. I think of the liver and diabetic dyslipidemia as very connected. I mean, the lipid is a major organ involved in lipid metabolism, production of VLDL particles, which are elevated in diabetics. The liver's full of fat and overproducing VLDL particles. The flux between triglycerides and cholesterol leads to small, dense LDL particles, which are atherogenic, and low HDL particles. And so what you see in the plasma in terms of the dyslipidemia is the tip of the iceberg, the liver being the iceberg. So there are abnormalities in triglyceride metabolism and obesity, type 2 diabetes, insulin resistance that lead to 75% of diabetics having the fatty liver, which also predisposes to cardiovascular disease. So most people with NASH, which is the inflamed part of fatty liver, actually, if they have increased cardiovascular mortality, and their greatest morbidity mortality is from cardiovascular, not liver disease, although they do have increased incidence of severe liver disease. And then the other thing that's interesting is that non-alcoholic fatty liver disease is associated very commonly with hypothyroidism. And so that also gives us a hint that our target might be important. Now, the progression from fatty liver to cirrhosis, it doesn't always occur. The prevalence of fatty liver is very high in the US, on the order of 25%. It's very prevalent worldwide due to the increase in obesity. It's estimated about 6 million in the US have NASH, and then of those, 600,000 with NASH-related cirrhosis, and there's also an increased incidence of hepatocellular carcinoma. So NASH, or NAFLD, is expression of the metabolic syndrome of the liver and is associated with all the other things that we think about as being metabolic syndrome components. The mortality in NAFLD/NASH is estimated based on data that's not all that good, and I'm sure that Manal could tell us more about that, but the 5-year mortality is about 67%. The 10-year mortality, or the survival, is about 67%, and the 10-year survival is only 38%. So you can tell that it's really kind of it's a marker of severe ongoing metabolic disease. And so our thought was that a compound that's efficacious in NASH and then hits other components of the metabolic syndrome, like LDL cholesterol and triglyceride and liver triglyceride lowering, has a significant upside potential on differentiation in what could be a very large market, given that there's no currently recognized therapy. The beta agonist, as you heard and John introduced, is trying to take advantage of the metabolic benefits of thyroid hormone without affecting the other target tissues. And what made this possible was the molecular understanding that thyroid hormone binds to different receptors. There's two major receptors, alpha and beta. The alpha receptor is the predominant receptor in all of the tissues except the liver, where the beta is predominant, and also in the central axis where in the pituitary the beta receptor is predominant. And so in the liver, the metabolic benefits are the lowering of cholesterol, triglycerides, Lp(a), and hepatic triglycerides. The undesirable side effects you heard about, tachycardia, bone loss, other systemic actions. And so the goals of this program, which really started when I was at Roche Pharmaceuticals heading up their CV metabolic group in Nutley, New Jersey, was to get a beta-selective agonist and then have it be targeted to liver uptake, so it wasn't getting into the brain and suppressing TSH, et cetera. And so how do you do that, is the question, and I'll show you a little bit about that. And then, what is the mechanism of these metabolic benefits? So thyroid, the beta agonist or thyroid hormone, which binds equally well to the beta or alpha receptor, is actually a transcriptional regulator when it's bound to its receptor and either activates or represses genes that have effects in the liver to increase reverse cholesterol transport. There's evidence that there's an increase in LDL receptors, but the dominant action is increased cholesterol metabolism. So it's really breaking down and eliminating cholesterol from the body. And then also to increase fatty acid oxidation and reduce the production of VLDL, as well as reduce hepatic triglycerides. It has, in addition, an anti-diabetic benefit and reduction of hepatic insulin resistance. And the LDL mechanism is distinct and complementary to statins, which reduce the level of HMG-CoA reductase while having little or no effect on the metabolism of cholesterol. There's validation for the literature validation of the beta target as well in NASH and NAFLD based on the genetics of patients with TR beta mutations, the RTH patients. They're insulin resistant and have fatty livers. As I mentioned, patients with fatty liver disease have abnormally low thyroid hormone regulated gene expression in their livers, indicative of liver-specific hypothyroidism. And then some T3 itself and some more selective THR beta agonists reduce liver fat in NASH in animal models, and then the knockout for TR beta have increased liver fat and delayed liver regeneration and repair. So this field has been plagued by molecules that weren't quite as selective as they needed to be, or studies in the past where thyroid hormone itself was actually used to try to achieve metabolic benefits, including weight loss in that case. And the toxicities were, the benefit was outweighed by the risk of these therapies. But nonetheless, there was clinical validation for the metabolic effects of cholesterol and triglyceride lowering. Clinical development was discontinued. The most notable compound was a proterome, which was developed by Carol Bio. And the reasons were that there were low margins, relative suppression of TSH in the central thyroid axis, some low-level liver enzyme elevations. There was an impurity in a proterome, a mutagenic impurity, and then a cartilage finding in a chronic dog study. And so the Roche program focused on identifying compounds with an improved safety profile. And this shows the chemical structure of 3196. It was discovered using a novel method of assessing receptor selectivity and activity. And so here, if we look at beta selectivity on the y-axis and then the potency at the alpha axis. So here, if you go to the right, it's less potent at the alpha receptor. And if you go up on the y-axis, it's more selective at the beta receptor. And the T3 sits here, right at the 0, point. It's highly potent at both receptors. It turns out that all the other analogs are also highly potent at both receptors. So even though they're a little bit better at beta, it's not enough of a difference that you still wouldn't have a major alpha effect in the liver as well as outside the liver. So this assay that was used was able to really tease out that functional difference more than prior assays. And that's something that I could talk to the biochemists among you about if you have an interest. So it's highly beta selective. We showed it was taken up by the liver, very low extrahepatic tissue uptake. It was the only analog that was negative in a heart assay that was designed to measure the kinds of negative effects of thyroid hormone on the heart. It doesn't get into the heart, and it also doesn't have the alpha activity. And in our phase 1 studies, we showed there were no effects on liver enzymes in healthy volunteers who have normal liver enzymes. And so this is one aspect of the biochemistry of how we used what's called a co-activator recruitment assay to show that we were very selective with this compound at the beta receptor. So the old compounds were all about like T3 in terms of their selectivity. That's 1x shown here. And this compound was 28-fold selective as opposed to 1, so 28-fold more selective. And then it also had almost no potency at alpha, so 3.7 micromolar as opposed to 3 nanomolar with many of the other compounds, which was very similar to thyroid hormone. And so comparing to T3, which is shown on the left, and 3196 is shown on the right, and what we showed here with T3, that different doses of T3 given to animals that they're called diet-induced mice. They're treated with a Western diet or high-fat diet. And T3 will lower the cholesterol in these animals, but it also reduces bone mineral density at every dose. So it's having adverse effects on the bone. It reduces T4, TSH. It also had other increase in heart size and at all doses, et cetera. Where there was no effect on bone mineral density of 3196, however, it lowered cholesterol extremely effectively in this model. Another study that shows a differential metabolic effect from T3, this time in a rat that has is high cholesterol, is that at the doses of T3 shown in blue, where you see profound reduction of circulating T4 and also of TSH, there's very little effect in reducing cholesterol and no effect in reducing liver triglycerides. So in order to have T3 actually be effective in the metabolic parameters, you're already seeing, TSH suppression and other adverse things. So you can't simply give T3 to correct liver abnormalities, and I think that's what John was telling you by providing the evidence on the clinical patient. And so you need a compound like 3196 where you can see that there's very, very little effect on T4, but a nice reduction of liver triglycerides and non-HDL cholesterol shown here. And this is what is a diabetic dyslipidemia model. It's just another model. These are animals that are very diabetic. Their triglyceride levels shown here on this graph here are 600 milligrams per deciliter. So this is a very hypertriglyceridemic animal model, and one in which you can show the effect of a compound on triglycerides. And here you can see that 3196 is able to reduce triglycerides by about 70% in this model. It also raises HDL very nicely, and will only do that in a hypertriglyceridemic model. Otherwise, it'll be neutral for HDL, but it's because of the flux between these particles that when you have very high triglycerides, you have low HDL, and so the compound can raise HDL in that case. And then reduction of liver weight, which is related to the reduction of liver triglycerides. And I just highlight again how the patients with resistance to thyroid hormone show some of these same effects where they have the fatty liver and insulin resistance. Now, the compound is very important in the lipid field that whatever compound you have works with statins. Many of the people that have metabolic syndrome or diabetic or whatever are going to be on statins. And so we did a study in a relevant animal model. It's a hypercholesterolemic rabbit model in which we treated either with atorvastatin shown in blue, 3196 shown in the lighter orange here, or the two together. And we were able to show that we had a nice, at least additive, if not synergistic effect of 3196 in combination with atorvastatin. A decrease in VLDL cholesterol and LDL cholesterol was seen with both of these drugs, and then again in combination. It also reduced triglycerides in this model up to 50%. And then finally, we did a series of studies in the diet-induced obese animals, both very short term, so we could see the immediate effects on gene expression. And then this one happens to be a 3-week study. And then we also did a 6-month study in this animal model. Now, this is an animal model of insulin resistance. So they don't really have frank diabetes. They do have very fatty livers. So the control on the left shows the vacuoles, evidence of hepatic fat. There is, so that's really the major finding, and they're insulin resistant. So on this panel, what you see is a reduction in liver size, which relates to the reduction of liver triglycerides. And that's also shown in this panel here, in which the amount of triglycerides in the liver is reduced dramatically. And I'll show you how that impacts the liver size in another slide or two. And then this was compared with rosiglitazone, which is a TZD, an insulin sensitizer, which has little effect on liver size or liver triglycerides, but it is a nice insulin sensitizer shown here in blue. This is an insulin tolerance test, and the 3196 worked almost as well as rosiglitazone in terms of insulin sensitivity, and we believe this is because a fatty liver is an insulin-resistant liver, and so you're reducing fat in the liver and improving insulin sensitivity. At these doses, the compound was weight neutral, or a slight weight loss was observed. Okay, so this is a slide and it's way too complicated, but I'll try to point out what is interesting here. This is the 6-month study, again, the diet-induced obese mouse. And in this study, we also had the lean controls. So if you look at just body weight on this left panel, the lean control body weight is shown in light brown, and then the diet-induced mouse body weight is in dark brown. So there's an increase in body weight with a diet-induced obese, in the diet-induced obese animal with the high-fat diet treatment. And then at the dose of 3196, which is very similar to the human dose that is efficacious, there's almost no effect on body weight. A very small reduction is seen. If we look at the liver weight, it goes up as well in the DIO as compared to lean mouse. The liver weight is completely normalized by 3196, so it goes back to normal. Again, rosiglitazone, that doesn't happen. And so what we're saying is that if you think about these animals, they're really overweight. So think of a person that is overweight and you're treating them with a drug like this. They have fatty liver. What you would expect is that you'll get reduction in hepatic fat, and at the dose that we're using, you won't see an impact on body weight. It might be a minor impact, and the impact, if anything, will be because of increased energy expenditure. So the drug used at the doses we're expecting to use will have a very minor increase in energy expenditure. TR beta is a nice effector in brown fat, and there's a lot of interest in humans' brown fat these days. And TR beta in brown fat, once it's outside the liver, will dramatically increase energy expenditure. So this shows on the right panel, if we use the 0.3 mg per kg, which is the light sort of orange here, you see just a small increase of energy expenditure above this line. But as you give 10 times more, you see a 26% increase in energy expenditure. It's a very dramatic effect. And at that point, you will see some significant reduction in fat mass and body weight. So this is not how we plan to use the drug, but we think a small effect in body weight will actually be beneficial. And so here is data related to the Nash and fibrosis pathway genes in the liver. So we noted so we have gene arrays from several of our experiments that we did in animal models. And we noted that the diet-induced mouse, shown here, has so red means increase. So it has an increase in many of the transcripts that are connected with inflammation in the liver and fibrosis in the liver. So an increase in galectin-1, an increase in collagen-3, for example. And that shown here is treatment with 3196, which the blue means it's reducing all these transcripts. So we get upregulation in the chronically treated, 6-month treated, diet-induced mouse that's reflective of Nash, and that we get downregulation of these key Nash inflammation and fibrosis pathway genes by 3196. We also saw a reduction in ALT, and interestingly, the maximal effect was about the 3.3 mg per kg dose, and this drug exposure correlates with our 80 milligram human dose, and that is the dose where we also see the near maximal effect on the lipids. So we think we have a nice link between the dose in the animal and then the dose in the human in terms of thinking about future studies. So moving now to the clinical study that we did, we've completed, as I mentioned, some phase 1 clinical trials in healthy volunteers, a single ascending dose, a multiple ascending dose. We dosed up to 200 milligrams for 14 days. The only criterion for inclusion was that these were healthy volunteers. They had to have a slightly elevated LDL cholesterol, greater than 110 milligram per deciliter, which is not very elevated. But for this purpose of this study, which was done primarily in college students and people living in Fargo who were relatively healthy, it was about 50% of people that had a screening that was done in which the LDL was greater than 110. So that was a good way to do the study for us, and we knew that we could see an effect. So there was well tolerated, safe at all doses tested. There were no events at the top, no adverse events at the top dose, so low number of mild adverse events that were not dose related. The drug exposure, as I'll show you, was dose dependent, and the lipid effects were beneficial and rapid. And this is phase 2 enabling studies have been done. So it's hard to see the PK here, but there's a dose-related increase. For the 50 to 200 milligram doses, day 14 over day 1 exposure shows ratios that increase slightly. This is what we expect for a drug that's taken up into the liver. So what you see here is the plasma levels of the drug. It's not reflecting what's in the liver. And the fact that there's a slight increase in exposure at day 14 to day 1 suggests to us that the half-life of the drug in the liver is much longer than the half-life in the plasma. And so we expect to be able to dose once a day, oral dosing once a day. And this is something that we would want to explore more in subsequent studies to understand a little bit more about the PK of the compound. There was no effect on vital signs in the study, maybe a slight decrease in diastolic blood pressure, but that's not a bad thing. And this shows heart rate on a Holter monitor. The top dose is in orange, shown here, which is basically lying right on top of the placebo, which is in purple. And you see the diurnal variation of heart rate. The highest peak is at the lowest dose, so there really was no effect on QT, et cetera. The one effect that we saw is on the free T4 level at the highest dose, and I will explain that in a second. The free T3 level, which is the level of active thyroid hormone, was unchanged. There was no effect on TSH at any dose. And again, this shows diurnal regulation of T3. And then the free T4 level, T4 is normally converted to T3. In the liver, it's converted to T3 by what's called deiodinase 1. And we know that our gene will upregulate deiodinase 1. So we expect, because it's a liver effect, that we will see evidence of T4. T3 will remain normal because T3 is really regulating the TSH level, but there's about a 20% decrease in free T4. Is still in the normal range, which is shown here. And I'll show you that if we're at the 80 milligram dose, we don't really see much of an effect at all. So this is a reversible, of course, and there's many drugs that are commonly used in which T4 clearance is affected as well. These are the lipid effects that we see in the 2-week study, And their cholesterol, LDL cholesterol, ApoB, non-HDL cholesterol, and triglycerides. So the maximal, as I mentioned, maximal effect is seen on this bar here, which is about 80 milligrams. And there's not much change between 80 and 200. 50 is somewhat lower than 80, but we only had one sort of non-responder in the 50 milligram group. So we feel, it's highly dose dependent, and we had about up to 30% reduction of ApoB, total LDL, non-HDL cholesterol, and then the triglyceride reduction was quite significant in terms of the magnitude, up to about 60%. But there was only trends because there's only 6 subjects per arm, and the triglycerides were more variable, and none of these people were really hypertriglyceridemic. They became more hypertriglyceridemic in the study because they're sitting at bed rest for 2 weeks, and that didn't happen in the drug treated. So the placebo became more, you can see the triglycerides went up. This is, this is corrected for placebo, but this is the lowest dose. So there was a non-effective dose. The triglycerides tend to go up, and then as you give the drug, they actually went down. Okay, and then here's the time course of effect. So we're giving the 14 doses, and then we stop the drug, and they leave the unit. And so what happens is the LDL, the ApoB, the non-HDL cholesterol all start down. You start to see the separation about 2 days after dosing, and the maximal effect is about 8 to 11 days. So it's a very rapid effect, which makes it something that's quite nice to study in terms of plans for your clinical trials and monitoring the effect of the drug. So in summary of this part of the talk, we feel this is a best-in-class thyroid beta agonist. We have proof of concept based on the phase 1 for the LDL effect. There seems to be an effect to lower triglycerides, which will be better established in patients with higher levels of triglycerides in the phase 2 study. The safety profile was excellent. Particularly not seeing any ALT elevations at all. That had been, minor elevations had been seen with all the other prior compounds. And then we have a strong set of preclinical data that support the lipid and NASH indications. Now, if we look at how do we take this drug ahead, you all are aware, how difficult it is to compete in the space. Dyslipidemia is very highly competitive. There's generics that are not very expensive, both in the LDL arena and the triglyceride arena. And so there, and increasingly, I would say, there's a requirement for CV outcome studies. This, when the first LDL therapies the statins were launched, they didn't have to study that many patients. And there really was no knowledge of whether they were showing benefit for several years after they were already on the market. But that's not the case now, and I would say the trend is going in the other direction. Great to hear your input on suggestions as to how to avoid this issue, but we think that the NASH regulatory environment is improving because of the high unmet need, and this could be more of a fast track to registration, possibly. So if we look at all the ways we could take this compound ahead, we have the brown path, which is an orphan path. We have sort of the blue path, which is the sort of mid-range, development costs in type 2 diabetics with fatty liver or NASH. And then the pink path, which is the megabuck path, where we're treating LDL cholesterol in combination with statins. Of course, we're going to hit the triglyceride endpoint as well, but a major market once that would be approved. The stage we're at now, the things we have to do are largely shared by the options. So we can design a study where we can understand better the effect of the drug, we have to do certain other parallel activities like the manufacturing studies and the formulation studies and the toxicology studies. And that wouldn't matter how we did the clinical design. So what we're thinking about is what is the best clinical study, the phase 2A study, so we can better understand the action of the drug. As I mentioned, looking at the NASH potential, there's currently no robust therapy for NASH. Lifestyle modifications, some drugs that have shown some benefit, like vitamin E, et cetera. But a lot of off-label sales, people are struggling with this. People come in, they have elevated ALTs, they have diabetes, what do you do? And there's the repertoire is not that extensive, and most people can't. The lifestyle is a difficult one. So the regulatory. So I've been sort of watching this. I've been talking about this since 2006 as a potential target. This beta agonist, a potential target for NASH, and the regulatory environment has just been like, well, we don't know what to do. We don't know what the endpoints are. The Clinical Research Network in NASH was established, I think, more than 10 years ago. You have that here at Duke, sponsored by the NIDDK. It's really built the knowledge of the NASH area and really helped define the entity, I think, and brought it to the light. Brought it to light and so that energy, I think, has really helped move things. There was a recent workshop in September of 2013. The FDA is beginning to listen, and we hope more than beginning to listen. And so what are the markers, and how do you develop a drug for NASH? There's some recommendations about the early stages of what one can do non-invasively to look at likely patients who might have NASH. It seems that there's going to be a requirement for serial liver biopsies, which itself is a surrogate in some ways. The liver biopsy is a surrogate for improvement in mortality and morbidity. And of course, we're going to want to see what the drug does in the cardiovascular arena because, as I mentioned, most NASH patients die from cardiovascular disease. So right now, what's been established is some histological scores that are fairly well agreed on. The Nash score, or NAFL activity score, which looks at fatty liver, ballooning in the hepatocytes and evidence of inflammation, and then the fibrosis score, which looks at the more severe components of cirrhosis, of beginnings of cirrhosis, and how that's scored. These are drugs in development for NASH. There's a number of different pathways. Just to highlight, there was recent news about Intercept's drug, obeticholic acid. Duke was involved in some of the studies of that drug. And it showed benefit in a phase 2 study in terms of reduction of the score. The data has not really been released, just a sort of a press release. They stopped the study early because the drug showed benefit in a serial liver biopsy at 72 weeks. It shares some common pathways with thyroid hormone. However, a downside of this drug is that it actually elevates significantly LDL cholesterol by 20%. And then there are some other, this drug from Gilead targets a collagen mechanism, so it's more of an antifibrotic. It doesn't have, if we differentiate it from our drug, it is an antifibrotic that doesn't address the underlying metabolic pathophysiology of NASH. And so our thought is that a phase 2A can establish safety and efficacy in this indication to support later development. And this is how we see it fitting in to the NASH treatment paradigm. So where will 3196 fit? If a patient comes in with metabolic syndrome, fatty liver, persistent elevation of ALTs, which are not from other liver diseases, their PCP could do some of the workup and follow these patients with lifestyle modification and treatment of metabolic risk. However, persistent ALTs and other highly suggestive biomarkers or clinical syndrome highly suggestive of NASH rather than simple fatty liver will lead to a liver biopsy. If the liver biopsy shows simple steatosis, then the metabolic risk is treated. If it's NASH, then hopefully there'll be a treatment for NASH. So the idea would be that 3196 would be developed to treat the NASH, but then because it has these advantages in metabolic, lowering cholesterol, triglycerides, whatever, it will start to be used earlier by physicians at this stage where they're not sure yet if they have NASH, but they do have the metabolic risk, or it will be treated or used in the simple steatosis, again, to treat the metabolic risk to actually prevent the progression to NASH. And to do that, it would have to be post-NASH approval and with additional safety and large safety studies, hopefully post-approval, and maybe even CV outcome post-approval. So this, I don't want to spend too much time on this. We really are just starting to have discussions about, well, what would a phase 2A proof of concept in NASH look like, and could we use that trial to get to also understanding better the lipid effects. And some of the criterion we've thought of, and again, we're discussing this with people who are really expert in this area, is who are these patients? We believe this drug is a good drug for diabetics. So insulin resistant, diabetic, with NASH, diagnosed by liver biopsy, because that's the only way you currently can diagnose NASH, and maybe on a statin, maybe on metformin, and then treat 6 to 12 weeks. We think that the 80 mg dose is going to be the right dose for NASH, but we'll probably also want to treat somewhat higher just to explore dose range. Do serial liver MRI or tracer for triglyceride production or biomarkers that shows that we're really getting reduction of triglycerides in the liver, monitor lipids, all the safety measures, biomarkers, CK18, ELF is something that was used initially by the Intercept drug, which is a series of biomarker panel, diabetes endpoints. And then we can, out of this trial, we'll get some evidence on the robustness of lipid endpoints, maybe some combination data on statins, although we may need a formal drug interaction study with statins before we dose with statins, some assessment of insulin sensitivity, and then are we getting evidence predictive of efficacy in NASH? And this would be, kind of what I mentioned as our development plan. We're here, thinking about a 2A study, a 2B3 study. Hopefully the same patients that we treated in the 2B could also become part of the phase 3. I mean, it's a pretty significant treatment for phase 2 that you have to treat for 72 weeks. So hopefully, that could be incorporated in the registrational study. And then, as I mentioned, this sort of CV and safety extension. Okay. Now, So this is, where I would like to stop, and then just open it up for questions. And I think there's supposed to be one last slide, but I can't get to it for whatever reason. I don't know. It's stuck. But I want to acknowledge all my collaborators at Roche and Madrigal and the various people that we worked with over the years. So thanks very much.
Thank you very much. That was great, interesting science and development pathway. So I have a question about the liver. I mean, it seems like this drug is mostly targeting. I mean, its uniqueness is really targeting liver fat, and most of what we measure is plasma lipids. And so, is the time course of effect on the liver and the durability of effect on fat in the liver similar to that in plasma? I mean, the
Right.
data you showed us is
So
really plasma.
what we can say so we don't have data on liver effects in humans, on liver lipids in humans. So what we can only say that the time course of what we see in the plasma is very rapid, right? But in the animal models, we have a major effect on liver lipids within 3 weeks. So if humans are like animals, then we would think it'd be fairly rapid as well.
And durability? I mean, so if you treat people for 6 weeks, how
Well,
long?
so the animals was very
How long does it
durable.
take them to reaccumulate all that fat and all the bad effects of that fat in the
If you
liver?
took them off the drug,
We don't know.
we've never done that. I don't know. I mean, I think that there was some durability just with our 2-week study when we removed the drug and then went out a week later, the plasma lipids were still impacted. So there was some durability there. I can't tell you anything about the liver, but that's a good question. Yeah.
Because and then the other question I don't know if others in the audience, Jennifer, might know, is whether people with we have these new lipid guidelines out, and whether people with diabetes or with in whom I sort of know the answer, or with NASH, how many of them are treated with statins, and what are the effects of statins on liver lipids? Because again, that's sort of the unique effect of the drug.
Yeah, well, I mean, Manal, maybe you could just, because, I mean, she's the person treating.
We use statins quite a bit, and there's actually some data, although it's not yet published, that the effect of statins on fatty liver, in fact, actually improve liver aminotransferases and have had positive endpoints, improvements in the endpoints on histology, both steatosis and inflammation and fibrosis. So the statin seems to be having more effect on liver injury beyond lipid lowering alone, and maybe functioning as some anti-inflammatory type properties, as it has in other areas of literature.
Yeah, but also, it was mentioned that when people have ALT elevations, their PCPs don't want them to be on the statin. So, we would agree. I mean, we don't believe that statins would contribute to injury, and they do have, like they lower in CRP. That's one of our markers. CRP is elevated in NASH. So they do have this anti-inflammatory effect. But I think it's interesting that, you mentioned that we might be able to find patients who are not on statins because of this persistent concern that, if people have ALT elevations, the docs don't want to put them on the statin.
I've always been very intrigued with the whole lipid issue in liver disease in and of itself, because when the whole story of fatty liver disease initially unfolded, everybody was harping on the insulin resistance and the diabetes and issues related to glycemia. And it turns out that as time has gone on, we've associated insulin resistance with fatty liver disease, and it's certainly necessary for the disease, but not required for disease progression or resolution.
Right.
And the PIVN study showed that you can improve insulin resistance, but not change the liver disease, and some patients may continue to progress. And even if you put patients on vitamin E, you can have worsening as time goes on, insulin resistance, and yet improvement in liver disease. So the whole story of insulin resistance, I think, is unraveling, and on the forefront is the mechanisms pertaining to lipid metabolism, and the secondary inflammatory effects of excess triglyceride or free fatty acids or whatever it may be in visceral compartments. That's one comment. One question is, as it pertains to your compound, what was happening with tissue concentrations of T4 and T3 T4 ratios? Do we know? Do you know? And
So
at the tissue level, are there
so
downregulation
the
of
plasma levels are normal. So TSH, all the thyroid measures are normal. So free T4 is normal, T3 is normal, and TSH is normal. So given that, there's no reason to think that there would be a difference in the tissue level of T4. But certainly, if we were dosing, let's say, the 200 milligram dose, and we wouldn't want people to fall below a cutoff, a free T4 of a certain cutoff, let's say, 0.8 or 0.7 or whatever would be considered the normal range for that.
So I don't know about this, as you were asking about study design ideas and issues. The majority of our patients with fatty liver disease have the whole dyslipidemia syndrome, about 60% actually, the elevated LDL, low HDL, high triglycerides. And would the FDA allow you to go after a dual indication in the same study design? In other words, if you were to study dyslipidemic patients who also had the outcome of interest with two outcomes in mind, not only improvement in lipids from a metabolic syndrome standpoint, but improvement in the liver disease, if your population that you're evaluating has both outcomes?
Yeah, so the answer to that is, for a phase 2A study, we can do whatever we want. Okay, it's not registrational. There's an exploratory aspects to it. In terms of, if we were doing a pivotal trial, we really need to know what patients we're treating. And yes, so the answer is, and you'll have to have a primary endpoint. I'm not the clinical trialist, but I think John can.
The FDA is interesting because there's substantial heterogeneity across divisions in the FDA. And so I think the endocrine group in the FDA has been driven by their diabetes experience to want it starts with cardiovascular safety, but now, but there's an extension of that, recognizing that most patients with diabetes are dying of cardiovascular events, that they're also interested in cardiovascular efficacy. And I think when the endocrine division is now thinking about approving weight loss drugs or lipid lowering drugs, they're asking the questions about, okay, what's the cardiovascular safety and what's potentially, is there cardiovascular efficacy? And that's driving the large clinical trial sample sizes here. So I think, and then in GI with fatty liver, I'm not familiar enough with the regulatory pathways, but I think if you did what you suggested of going after these sort of two indications, you'd wind up getting involvement of two groups at FDA and probably a lot of complexity because of that. But it might, yeah.
Yeah, I was more thinking about, you kind of go for the first approval, and then you could hopefully go for something, while you're already approved after that.
Hi, and I'm Jennifer Green in endocrinology. I just wanted to follow up on the prior point. I think it's taken endocrinology to come around, a long time to come around to a point where we're not just focused on numbers looking better. And I think unless you were going to target a particularly underserved population, such as individuals with familial hypercholesterolemia, for example, it would be very difficult to get an indication for a drug to simply make lipid numbers look better. I think, we're past that. And it's really going to they'll need to be, the reason we expect those numbers to look better is because we think that in turn there will be a cardiovascular benefit. So I think, if you're really going to target this as lipid-lowering therapy, there have to be some actual events, and an appropriately designed trial.
I was at the FDA meeting in 2013, and I think the FDA is also past that. So the FDA wants hard outcomes.
Great talk, thank you very much. I'd be interested, is there any differential response or differential effect if you're in mice or ultimately in humans that are given supplemental exogenous thyroid hormone?
So that was what I was trying to show, that in the early slides, and maybe I should have put them a little later, but that you can't give thyroid hormone to get the effect, because at the doses of thyroid hormone, we already see central axis suppression, and you're already going to see heart rate, and you're going to all see these bone effects. You can't separate the benefit in the liver from the adverse effects outside the liver with thyroid hormone. And it's also, I think there's another flag that I would worry about. I also think having an alpha effect, which, the thyroid alpha, there is thyroid alpha in the liver, and particularly in the cells that are called stellate cells that make the collagen and the fibrosis or whatever. So it actually could be an issue because thyroid hormone hits both receptors. So not only outside the liver, but maybe even inside the liver, we don't know what it would do.
Great.
Okay.
So in the interest, thank you very much. In the interest of
Thank
time,
you. Thanks,
we'll call
everybody.
it quits. Becky will be around for the rest of the day visiting with people, and then hopefully we'll have more collaborative relationships going forward. Thanks.
Thank you.